Good morning. My name is Natalie Artzi, and I'm an assistant professor at the Department of Medicine at Brigham and Women's Hospital. My lab is passionate about bringing new technologies to the clinic, and today I'm going to tell you about a polymer-based nanoparticle platform we developed for gene therapy for the treatment of solid tumors. Research in the past few decades has culminated in the approval of oligonucleotide-based therapies, cell therapies, and in vivo gene therapies using AAV vectors to treat diverse clinical indications, including neuromuscular disease, inherited blindness, and blood cancers. Beyond transgene delivery, gene editing technologies, including CRISPR, are enabling precise modification of human genome sequences. Looking into the future, personalized therapies and epigenome editing can impart sustained outcomes with the ability of tunability and reversibility. However, despite the rapid pace of technological innovation, a primary impediment to broader application is the challenge of nucleic acid delivery that results in premature degradation by nucleases and immune response to products of foreign transgenes. The other challenge is how to target tissues outside of the liver and retina where successes have been demonstrated, while eliminating off-target effects. While viral vectors used for gene delivery have found a lot of successes in some areas, almost 50% of patients treated with AAV-based therapies are currently excluded from treatment due to pre-existing immunity to the viral capsid. Although the vectors can be engineered to evade immune recognition, their high cost of manufacturing and limited packaging capacity fueled research in the area of non-viral delivery systems. Polymeric, metal, and lipid-based nanoparticles are likely to have tremendous impact on gene therapy, building on the clinical success of Onpattro, a lipid-based nanoparticle delivering siRNA for the treatment of amyloidosis in 2018, and more recently, the nanoparticles that serve to deliver mRNA vaccine against COVID-19. They're really built on years of research and development of delivery systems for the treatment of cancers. Nanoparticles have the potential to evade detection by the immune system and thus enable redosing, a limitation of viral vectors, and can be engineered to add targeting agents and functionalities that can help with gene therapy outcomes. The other advantage is that those particles can be engineered to deliver a range of nucleic acids that vary in their physical and chemical properties. We and others have demonstrated successful delivery of plasmid DNA, mRNA, and RNAi using polymeric particles via, for example, self-assembly of positively charged particles with negatively charged nucleotides. However, the delivery of small molecules such as cyclic dinucleotides or CDNs have raised a lot of interest as cancer vaccine adjuvant is more challenging because CDN small size does not allow for the formation of a stable complex and require more sophisticated design of nanoparticles to shuttle the molecules through the bloodstream to the cell while reversibly releasing the cargo in the cell cytosol. Nanoparticles that were traditionally designed to deliver cancer cells killing agents are now designed to target immunomodulatory factors to immune cells in order to generate a robust and long-lasting immune response against tumors. So why the interest in immunomodulation and in CDN in particular? In 1891, William Coley, the father of immunotherapy, began using pathogens to treat patients with unresectable tumors when chemotherapy and radiation were not available. He treated more than 1,000 patients, of which several hundred achieved near complete regression, but he couldn't explain why. It turns out that the innate arm of the immune system recognizes pathogen-associated molecular patterns, or PAMPs, through pattern recognition receptors to initiate an effective immunological response upon invasion of bacteria or viruses to our body, and that the activation of the innate immune system is an important driver in priming an adaptive immune response as well. CDNs are potent immune activators that activate interferon factor 3 upon binding of CDN to STING that results in the endoplasmic reticulum, which is a potent inducer of type 1 interferon. The uptake of CDN by antigen-presenting cells, including dendritic cells, activate them to present tumor antigens to T cells that become cytotoxic and can kill cancer cells. The upregulation of PD-L1 on cancer cells can prevent this interaction from happening, but the combination with checkpoint blockade therapy can help the T cells kill the cancer cells and eliminate it. Why nanoparticles? The high hydrophilicity and negative charge of CDNs really hinders the delivery into the cells. Our CDN conjugated to polybeta amino ester nanoparticles is now positively charged and enables systemic delivery of CDN that otherwise will clear from circulation within less than 30 minutes, and the particle is programmed to release the CDN in the cell cytosol. We've studied the safety, efficacy, and mechanism of action of our nanoparticles in multiple mouse solid tumors. Here in a melanoma model, you can see that we could redose 3 times 4 days apart the nanoparticles, and while untreated control resulted in significant tumor growth, the free CDN showed similar results, and the same dose of CDN, when formulated in the nanoparticle, significantly inhibited tumor growth that also resulted in a high improvement in survival. When combined with checkpoint blockade therapy, we saw further tumor inhibition that resulted in 60% of the mice surviving. We saw similar trend in colon cancer with free CDN and untreated groups resulting in significant tumor growth. Nanoparticle-delivered CDN inhibited the tumor, and we could get 70% survival and immune memory in some cases. In a less immunogenic model of breast cancer, the nanoparticles still inhibited tumor volume, but the survival benefit was not as significant, though still improved about 15 days compared to untreated control. Overall, our nanoparticles demonstrated safety and enhanced uptake in immune cells, and in some cases really generated immune memory. It is particularly interesting in the context of immunotherapy to understand how different administration modes affect drug pharmacokinetics and pharmacodynamics and its accumulation at the different organs. This is because the tumor microenvironment, as well as other lymphoid organs such as the lymph nodes and the spleen, play a role in determining the therapeutic outcomes. To answer this question, we developed a few technologies that enable, in addition of the delivery of intratumoral or intravascular delivery of nanoparticles, to now deliver them locally via an adhesive hydrogel that can stick to the tumor and release the particles locally over time, or a polymeric microneedle patch that enables transdermal delivery of the nanoparticles. These material-based delivery platforms can facilitate the identification of the optimal combination therapy, dosing regimen, and route of administration as a function of the drug mechanism of action and target site. Given the promising results we obtained using our STING agonist delivery system, next we would like to test their safety and efficacy in large animal models and non-human primates on its way to translation, and leverage the ability of our particles to deliver combination therapies to further enhance outcomes. We anticipate that material science will enable the convergence of traditional oncology that really seeks to reduce tumor burden and release tumor antigens with immunotherapy that aims to restore homeostasis by enabling the efficacious delivery and precise modulation of the immune system. Our platform technology can be used to treat many other diseases, including autoimmune diseases and for tissue engineering applications, which are additional areas we are pursuing in the lab. Thank you very much for your attention.